Alright, so in this video I'm going to be going over auxin and the key things to remember in terms of auxin and plants for the purpose of this class. But a quick disclaimer is that I recommend watching this video after already looking at some of Mark's auxin material because I'm going to be going over a review sheet that I made that hits on some of the important concepts when it comes to auxin and the role that and the extent that we cover it in this class. But I don't really talk about any of the cool experiments that detail how auxin's role was discovered in plants. All right, so looking at things pretty generally here, we know that auxin is a growth hormone in plants, and we know that phototropism is a plant growth in response to blue light. So how do plants react when they detect that blue light is in their environment? And so really with auxin, I think there are three important diagrams to remember in terms of phototropism and plants' response to blue light using auxin. And the first is right here, and it's kind of looking at auxin movement at the large plant scale. And so this is the 212 version of the plant that we use, where the shoot is above the soil here and the root is below. And we know from kind of learning in class, right, that auxin always starts up here in the shoot. It'll travel down the root, kind of come up here, and then go like that. So this is the shape that auxin kind of takes when it travels down the length from the shoot of the plant to the root. And so then kind of looking at these questions here, we know that auxin moves from plant shoot to plant root. And so I posed the question, what about when this plant is flipped upside down? And it's important to know that that doesn't change anything and it's still shoot to root for that. Even when the plant is upside down, it'll always go from shoot to root. And we'll see why here in a second. All right, so jumping over to the second diagram now, we're going to look at how polar auxin transport is generated in the tip of roots when the plant detects blue light. So we can see here that blue light is over on this right-hand side, blue light's coming, the plant detects it on its right side, and so we're going to see how the plant responds. And so first, what is the name of the blue light receptors? Those are phototropins. And just right away from looking at some of Mark's material, which way will this plant move auxin? It will move auxin to the left. And then how does it do this? It does it via auxin efflux carrier relocation. And so what does that look on the diagram? All these blue things right here are auxin efflux carriers. But the ones in the tip are interesting because the plant can choose if it wants to have them just going straight down like this, which it would do in normal sunlight. But because we have blue light coming in from the right, the plant is going to shift all of its auxin efflux proteins over to the left. So then it can move auxin through these efflux proteins through the tip of the plant and down to the elongation zone on the left-hand side. And so we know auxin works to increase plant cell expansion in the elongation zone. And so which way will this plant bend? This plant will now bend to the right towards the blue light because we have more auxin going down this left-hand side of the plant. All right, so scrolling down here to this third diagram, we're going to see how auxin movement and how the generation of the polar auxin transport leads to different elongation in the elongation zone of plants. And it's important to remember that when we're looking at this diagram, we're really just talking about what's happening in the cells in response to auxin at the elongation zone of the plants. And so if we kind of follow this description that I have written over here on the right, we know that auxin is actually just a weak acid, and so it has two forms. Its acidic form, HA, and its basic form, A-. And so the acidic form of auxin diffuses through cell membranes, and we kind of see that right here, right? HA is diffusing through the cell membrane. Doesn't need a co-transporter, doesn't need a channel, anything like that. And so once the acidic form of auxin is actually inside the cell in the elongation zone, it will activate the proton pump, which kind of looks like this in the diagram. And what does that do to membrane potential? Well, if you have a hyperactivated proton pump, you're going to have more protons flowing out. That creates a more positive charge going out, and that leads to hyperpolarization of the membrane. All right, and so these membrane potentials do what to the proton sugar cotransporter in the K channel leading to solute something? They activate. And so you see that now there's all these protons on the outside, so they're going to come back through, help bring in sugar onto the inside, and it's also activating this K plus influx channel. And so that obviously we see now the sugar is going in, potassium is going in, and so that leads to solute import in this case. So now looking at number 4, how does this change osmotic potential? Well, import of solute would lower the osmotic potential inside this elongation zone cell. And so decrease here. And then water always flows from a high water potential to a low water potential. And so since we've lowered the water potential, water will flow in. And so that results in H2O import as well. All right, and so now we can see that we have, just to kind of recap, we have the acidic form of Auxin diffusing through the membrane, hyperactivating proton pump, which leads to membrane hyperpolarization. These changes activate these two things that lead to solute import, and then water follows. And so now there's more water and buildup of pressure in the cell. In the meantime, these enzymes called expansins cut things called quick links only at low pH. And so this is happening in the meantime because now that we've hyperactivated that proton pump, we have a whole bunch of protons out here outside the cell. All those protons lower the pH and activates the expansion so they can cut the quick links. And in general, this will cut the quick links that surround the elongation cell. So now that they're becoming fuller with water, they're free to expand and they're not kind of held in place by that rigid structure because the expansins have cut that. And looking at the last question down here, what drives auxin out of these cells? Well, we know that once the auxin enters the cell, it is in its acidic form, but eventually it's converted to the proton dissociates, and now it's in its basic form. And so negative charge, what drives auxin out of these cells? Well, we always know membrane potential, positive out, negative in, so it's going to be definitely charge, and then it is also concentration as well. So charge and concentration down here. And then one other thing that I should mention is that we noticed that when auxin entered the cell in its acidic form, it was able to permeate through the membrane with no needed cotransporter, no needed channel. But when it exits the cell here, we see that it needs this channel because it's in a different form. Now it's negatively charged and it needs this channel to exit. All right, so looking down here at the last portion of this worksheet, we see auxin and how it plays a role in gravitropism and plants' response to gravity. And so we know that if you lay a plant on its side, the roots are still going to want to go down into the soil and the shoot is still going to want to grow up, even if the plant is placed on its side and in the dark. So there's no blue light effects like we were just talking about in this scenario because it's dark. So we know the plant is going to bend this way and then this way, respectively, in the root and the shoot. And then we know a high auxin concentration leads to what cell elongation in the shoot, and that would be increased. That's what we just went over here, where we see that the water imports, expansions are cut, so the elongation is free to occur. And so, looking at the question down here that says which elongation zones have more auxin, well, we know auxin always starts in the shoot, and if the plant is bending this way, if the shoot is bending this way, we know that auxin must have been sent down here. We know auxin doesn't like crisscross when it goes down, so it'll go down straight down this side and come back up. So these two zones down here will have the highest concentration of auxin. Okay, and before we move on, let's take a look at this question down here. So based on the graph, based on this graph, high auxin concentration leads to what cell elongation in the roots? And so this graph is kind of interesting, and we can think about it like if this is the normal concentration of auxin just in a straight up plant growing in uniform light, it's receiving equal elongation in the shoot, equal elongation in the root, and so it's just going to be growing straight up and down. But so now let's say we tilt it on its side and in the dark. And so we increase the concentration of auxin on this one side, right? Well, we can represent that in the graph here. So if this was the original auxin concentration, now say this is the new auxin concentration. Well, we see that by going this way, by increasing the auxin concentration, and we look at the shoot curve, we see that we are now at a higher spot on the shoot curve, and that corresponds to a higher elongation rate. And so from here we see that increasing auxin concentration in the shoot results in more elongation. We already knew that, but now let's see what happens in the root. And so it's really an opposite effect, right? Because this is the original auxin concentration line that crosses right here on the root. So you have this much elongation in the root with this concentration of auxin. But now we've gone way over here. And so this is our new concentration of auxin, and that doesn't even show up on the root elongation rate line. So that means that higher concentration of auxin in this scenario leads to decreased elongation in the root. Now it makes sense, right? In the shoot, we said more auxin leads to more elongation, so you'd curve this way. Well, in the root, you get less elongation here, which would lead to bending of the plant this way. And so, kind of just to wrap it up, auxin is an excellent example of a hormone, being that the same molecule can have very different, even opposite responses depending on what kind of cell we're talking about. And here we talked about shoot cells versus root cells in plants. I hope this review is helpful, but if you have any more questions about auxin or anything in the class, feel free to reach out to me, Mark, or any of the other TAs. I know we all love teaching about auxin, and it's our favorite subject.